Hi, everyone. So we're Maria and Noel.
Noel.
We are graduate students in the Mair Lab at the Harvard THM School of Public Health, and we're very excited to have you here and show you our lab. First thing I wanted to say is that please, any questions that you have about anything, just type it in the chat and we'll be happy to answer them. Or interrupt us anytime. Let's get started. And now we're going into the lab. This is our space.
So here's my desk space, and the first thing I'm going to do is put on my lab coat. And this is because safety always comes first in the lab. So putting on our lab coats helps us prevent any contamination that we bring from outside into the lab, and it also protects ourselves and our environment from any harmful chemicals that we may work with in the lab. In addition to a lab coat, you can wear gloves, goggles if necessary.
So what are you working on?
So in the Mair lab, we're interested in aging. And so what do you all think of when you think about aging?
Joseph or Envy, do you have any thoughts and what do you imagine when you think about aging or getting old? Well, I personally think about getting sick.
Yeah, definitely.
Cells dying.
That's how it sounds to us too.
Yes. So yeah, as we age, we have a more likelihood of being diagnosed with these chronic diseases like cancer. You may have heard of that. You also might experience you will experience wrinkly skin and some of those more cosmetic issues. So we're actually interested in looking at what makes the young and what makes the old. And to do that, we actually, if we did that in humans, that would take a lifetime, literally. So instead of researching aging in humans, we research aging in a worm called C. elegans.
You said a worm?
A worm, yes. So these worms are very helpful because they only live a couple of weeks. So we can actually perform our experiments on aging with a reasonable amount of time. So yeah, so I can show you where we keep the worms. So here's our incubator room. We actually keep the worms in this big machine that looks like a refrigerator, but it isn't. So here are containers that contain all of the worms. They actually like to be housed at 20 degrees Celsius, which is a little bit cooler than room temperature. So this really mimics the environment that they live in the wild. We can also keep our worms at cooler temperatures, such as 15 degrees, if we want to, say, have our worms grow a little bit slower than normal. So you may have noticed in those containers little circles, and those circles are called petri dishes, and that's actually where we keep the worms.
So how big are the worms?
So the worms are about 1 millimeter in length as adults, and we actually use our microscope in order to get a closer look at our worms. So within this petri dish, you have E. coli bacteria, which is essentially food that the worms really enjoy eating. And you also have on the outside, this more clear material is a nutrient growth media, which is essentially a mix of solutions that helps the worms grow. So to study these worms, all we need are these petri dishes, a microscope, a pick in order to manipulate the worms if we want to move them from one plate to another. And we also need a flame in order to make sure that we're not introducing any contamination within our petri dishes. So you may be wondering how we study aging in worms. And here are some worms at different stages. So as you can see, the answer's here, but the old worm is the one on the right. So by eye, you can really tell by the worms how they look, how old they are.
Are there any questions?
So even though by eye, these worms look drastically different from humans, 83% of their DNA or their genetic material is actually similar to humans. And this makes them relevant for us so we can learn more about their genes, which will inform us about our genes. So now Maria will show us how she studies genes within these worms. Yeah. So Maria, what are you going to show us?
So the first thing that we have to do is to collect the genetic material from those worms. So what we do is we pick them, as Noel showed you, and we put them in these tiny little tubes that have a liquid that is going to open them and let all the DNA, which is the genetic material, outside. Then we are going to use this machine right here, which is a thermocycler. And this is going to help us to expand the piece of DNA that we want to see. So it's gonna go through many different cycles. And from one little piece that you started with, you're gonna get many, many, many copies. And that we're going to be able to see it in a gel. But the first thing that we need to do is a way to separate which genes we have changed and which ones are the normal ones. So what I'll show you today is one in which I added another piece. I added protein that shows fluorescence. So that makes the gene bigger. And to separate those, we use these gels right here that separate this little substance. It's going to run and run, and then the big pieces are going to stay up and the small ones are going to go down. But let's actually see that.
Great question. So in this lab, we actually do study other animals. So in addition to worms, there's mice, and they are very similar, even more similar to humans. And we do have we do perform some experiments by aging mice to determine what is different in an aging mice versus a young mice. So that's a great question.
So then I took that gel that separated the genes and put them right here in this machine. You can see that's how it looks. That's the purple. And then we're gonna take a picture. So to be able to see the DNA, we mix it with a substance that it's going to get into the DNA. And when we put it with UV light, just like the sun, for example, but in a very, very large concentration, we're able to see the reflection. And this is what we see. So I have this right here. It's a ladder. So these are genes that I know which is the size of each one. And then I have 4 samples. So I wanted to see which of those worms got the protein added that I wanted. So which one do you think is the one I want? Which one is the bigger gene? in 1, 2, 3, or 4. It might be a little bit hard to tell, but if you go closer now,
We have one in V says 2
2.
is a bigger one. Yes.
Yeah, that's correct. So actually, 2 and 4 are the same, and they are both the ones that are the biggest gene. They are a little bit up than the other one. So I know that these 2 worms and the progeny that came to them are the ones that have that protein that is going to fluoresce. So I would be able to see it with my own eyes, as we are going to show you now. This is all of our lab. These are some of our colleagues. Now we're going to a special microscope room to show you in more detail how we change the worms.
So now I'm going to show you some worms closer up where you can see them with your own eyes. So first I'm going to put gloves on in order to make sure I'm having all the safety regulations. Also, I'm going to wear glasses in order to make sure my eyes are protected from anything that could potentially be on the eyepieces. And I'm also going to turn on my flame to make sure that when I open the Petri dishes, no contamination gets within. So the first worms you're going to see are wild type, which basically means worms that we all consider normal. And I will show you now.
From our gel, that would be the lane, the first and the third lane, that they didn't get any modification to their DNA. So these are totally normal and are going to be the control for us to see if what we did to the worms make them live longer or live less. Right here.
So these worms are actually hermaphrodites, which essentially means they have all the organ systems to make their own progeny. So one single worm can produce eggs without being in contact with any other worm. So they have both male and female parts, which makes them great because we can grow them in large populations to study them. So that's one form of the worm. We also have males, which you don't see very much in the wild, but we can make them in the lab. And I will show you that. So here is C. elegans. And I don't know if you all can see it. Does anyone see any differences from the first worms that I showed you?
Yeah, these look longer in size. I mean, in length.
Yes.
Yeah, so that's exactly correct. Males are longer. They also move a little bit quick, well, a lot quicker than hermaphrodites. And they do have on their tail, they have a bump, which signifies that they're males. So that's how we can tell under the microscope what's a male versus a hermaphrodite. So now I will show you an old worm, and you all can tell me what differences you see compared to the worms I previously showed you. Get big screen. Can you make it large screen? So what do you all notice in comparison to the worms that I just showed you.
When you think about getting old, how similar do you think that would be to what has happened to this worm? It's a little thicker, yes. We start to gain weight actually when we grow old. What about the movement? It doesn't move at all, exactly. You're totally right, Andy. So one of the changes that we see is that it's really slow or it just doesn't move at all. That if you think about it, it's very similar to what happens to us as humans as we grow old and old.
Okay. So now we're gonna show you how we can really track the genes, sort of like the genes Maria mentioned earlier. How can we track them? So as Maria said, she added a certain protein to the gene that she was interested in. And I'm going to show you what the effect of adding a protein can have. And we might want to turn the lights off for this one.
So we're going to use a special light under the microscope to be able to see the fluorescence that comes from the protein that I added to that gene. In this case, I tried to add a protein that glows green. And this is how it looks under that light. Can you see where the fluorescence is?
Yes, very cool.
Do you notice anything else about the movement of this worm compared to the others we have shown you? Think about where is it going? So this one, if you really look at it, it's moving in circles, exactly, Andy. So this one, in addition to adding the green fluorescence, we changed a gene that helps the worm move normally in a sort of straight line. This one, because we changed that gene, is just gonna go in circles and circles.
So there are other different ways that we can change genes within worms so that they appear differently. And I'm going to show you another one now. And we actually don't need the fluorescent scope for this. So we're just going to use a regular light to look at this worm.
Would you like to ask anything? Just put it in the chat or unmute yourself.
Okay. So do you all notice any differences compared to what we've previously shown you? When you look at these worms, what do you see?
They're shorter than the other ones, exactly. So they're actually shorter, they move slowly because it's like harder for them to move because they are also more thick than the other ones. In this case, we changed a gene that helped them organize their cells in their body. And that's why they just look a little bit chubby, a little bit shorter, and that makes it for them hard to move. This is another way we can know what's happening. So does this mean that older worms are thicker and don't move at all? It is one of the changes that we see when worms grow old. they tend to gain more fat. So they become a little bit thicker and they don't have as much energy as the young ones have. So that's why they also move slower. So what we try to do is to change genes and see if we can prevent that from happening.
So one interesting thing about selection is you can actually alter the food, the E. coli that they eat, in order to change their genes. So I'm going to show you now a population of worms that have normal E. coli and another population of worms where the E. coli are different in that they contain the genetic material to alter the worm's genes, which changes the way the worm behaves. And we can track that with the fluorescent protein that Maria has already spoken about. So here's a normal worm on normal bacteria. And yeah, we might want to turn the light off. Okay. So Maria is actually going to take a photo of this worm so we can compare it to the worm that is on the other type of bacteria.
Okay, let's try to catch another one. Great. Yes.
Okay, perfect. Now I'm going to choose a different plate where the bacteria is different. And the worm is the same type of worm, just the bacteria is different. Okay, let me focus. Okay, so let's look at those pictures that Maria just took,
This is the one we just took.
which has a different type of bacteria. Maybe I can show that one.
And this is on the right, we have the normal worm. Want to tell us what's different?
It may be a bit hard to see here, but we can see that by eye, the protein that was connected to the gene that we were interested in, what is
You're also right. The movement is
The
different.
movement. The movement is also different. So the fluorescent, the green that you see here is localized to a different region of the worm when we place it on a certain type of bacteria that introduces a mutation within that worm. So yes, and this is how we can use these fluorescent proteins in order to study the genes.
So that is a very brief summary of how we study aging in our lab using worms. I hope you get as excited as we are to be able to see these things with our own eyes, to be able to see the DNA, see fluorescence proteins, and to just be aware of how by changing one gene, the whole movement and the behavior of the worms changes. Do you have any final questions? No.
So thank you all for joining us for this tour today. It is recorded, so if you want to go back and watch it. Charlie will be following up with information about the recording. And yeah, this was great.
We're also going to put some information at the end of the video with some links to where you can actually contact us in case you keep thinking about this and have any question or would like to learn something else. It's really great to have you here, Joseph and Andy, and we hope that everyone that watches the recording also enjoys it a lot.